Hi everyone, Dr. Matt here, and in today's video we're going through the topic carcinogenesis, which basically means the generation of cancer. There's a lot to go through today. I'll point out what we're going to cover. We're going to look at some new terms, so sometimes there's complicated terms that are hard to understand what the differences are, so we'll go through those first. We'll go and compare what a benign tumour and a malignant tumour are and how are they different. We'll look at why certain tissues in the body are more prone to becoming cancerous. We'll look at the most common cancers both in the world and in the developed world, and we'll see the risk factors behind those. And then finally, I've created a mnemonic to help you with the 8 hallmarks of cancer. So these are the 8 traits that cancer cells must have to become cancerous. All right, so there's a lot to get through. All right, so today, let's start with some terms. The first term you may have come across is neoplasia. Neo means new, so this just basically means new cells plus their connective tissue. So when a whole cluster of cells start growing and their connective tissue around them, we call this a neoplasia or sometimes a neoplasm. So it's essentially a new growth. There's a new growth within the body. Now, sometimes this is synonymous with the term tumour. Tumour really just means swelling. It's a Greek word, so sometimes you may have come across that when you learn inflammation. It's a cardinal sign of inflammation, tumour. So technically it's a swelling, but most of the time when you hear tumour, it's a group, a cluster of new neoplastic cells. So when you hear in the context of oncology, tumour, it's referring to new group of cells. Now, when you come across this term, cancer, really you need to know that this is malignant. Now, why is it called cancer? Well, again, I think this goes to the Greek terms. So cancer comes from crab-like. And so you may have encountered this with the horoscopes. The cancer or the month of cancer is the crab. Why is it called that? Well, possibly the reason why it was named a malignant tumour, well, you've got the body of the tumour, like the body of the crab, and then you have these radiating features like the legs of crabs. And that may look like a crab if you're looking at it down a microscope or grossly, or it may have behaviours like a crab, which could be a little bit aggressive. But you should know that when you hear the term cancer, it refers to a malignant form of tumour. But a lot of the time for your patients, they will just use the term cancer regardless if it's benign or malignant. Now, before I get into the difference between benign and malignant tumours, swellings or groups of cells, a couple of further terms you just need to be aware of. You may come across the term hyperplasia. Hyper means increase, plasia means growth. So this is an increased growth of cells. Now this can be under normal circumstances. So if you have an injury or an infection, your body produces more cells to overcome that stimuli. That's normal, it's reversible, it will go back. So that's hyperplasia. Then you might come across metaplasia. Metaplasia basically means change in form. So this might be in response to a stimuli, and the cells change their form to try to overcome that stimuli. But again, this is reversible, they can go back. An example would be the esophagus in response to acid that's coming refluxing up from the stomach. Those esophageal cells may metaplasic, change their form to be better suited for that acid, but they can revert back to their normal appearance if that stress is taken away. Then we move to another term, dysplasic. Disordered growth. So what the cells are doing here is they're changing form. They're becoming a little bit disorganized, but they're not tumors, meaning they are still bound within that area and they're not grouping up as cells. They are still sparse, but they are disordered. Sometimes dysplasic cells are precancerous, so they may progress into cancer, but they are not cancerous and they are not a tumor. So now I want to go in and see how these two terms are different. So this refers to tumour, a swelling, a group of new cells, which could be a neoplasm. But what's the difference between this neoplasm being benign or being malignant? All right, the first characteristic is their differentiation, meaning are the cells in this tumour or neoplasm or the neoplastic growth, are they representing or are they looking like their neighbouring cells? If it's benign, yes, they are differentiated to look like that area. So if it's a skin tumour, if it's a neoplasm on the skin, if it's a benign, it would look similar to the neighbouring cells. Where a malignant, no, they differentiate, means they change form away from once what they originated from. So their differentiation is not like the neighbouring cells. So they actually de-differentiate as malignant, whereas benign cells stay differentiated. The rate of growth of benign cells is slow, where for malignant they are generally called erratic. So they can go through phases of slow and then fast, so they can actually speed up. Generally speaking, you would say malignant tumours are faster growing. In terms of invading the local tissue, benign tissues do not. They stay fairly well encapsulated and localized, whereas malignant cells do. So they do indeed invade, and that explains one of its qualities. And then finally, metastasis, meaning spread into somewhere else in the body. Benign, no, they do not. Malignant, yes, they do. And this is why it makes these ones so aggressive, because they have the ability to invade and to leave the area. Now, you would think straight away that malignant cells or malignant tumours, therefore, are the dangerous ones, which is true. These are the aggressive ones. These are the ones that are most likely to cause bad outcomes and death. Benign, a lot less likely to, but these can still cause serious problems, particularly if they're in areas of the body where we can't have these tumours to keep growing. Like brain tumours, there's not a lot of space in the skull, so if they start to increase in size, they'll take up room and start pressing on important structures, and that will cause dysfunction. Sometimes these benign tumours are from cells that produce hormones. So if you start to increase the number of the cells through a benign growth, and you start producing lots and lots of hormones, such as the hormones in the adrenal medulla, which produces adrenaline, you're going to have crises through too much adrenaline. So it can actually be life-threatening as well, but the malignant ones are much more likely to be dangerous because of all these qualities. Now I want to move you on to why do certain cells in the body are more likely to become carcinogenic or more cancer-like. And this comes down to tissues in the body which we call labile tissues, which means tissues that are constantly replicating or mitotically active, or their cell cycle is always turned on. These are areas of the body where you need to renew, you need to replace, recycle, refresh tissues such as the epithelium, blood, things like that. Other cells in the body, like stabile cells, these would be certain visceral locations like the liver, they are almost turned off or they're hibernating. They're not actively replicating. They can become or switch back on, but generally they are hibernating and turned off. So they are less likely to become cancerous because we're not changing the DNA, we're not replicating, therefore the mutations in the DNA are less frequent. And then lastly, we have permanent cells like neurons, like muscle cells. Therefore, because they are completely turned off, they're not going to replicate again, it's less common that you'll see cancers associated with that because we're not replicating these cells. Therefore, the likelihood of them being switched on and continually on is lower. Not to say the cancers don't happen there, but they're a lot less frequent. So the cancers will happen in the labile tissue. Examples would be the stem cells in the bone marrow. So these would be the hemopoietic stem cells that make the red blood cells, the white blood cells, the platelets. You're making millions and millions and millions of new blood cells per second. So there is likelihood or chance that these cells could go a little bit awry and then cancers can develop. And this would be things like leukemias, lymphomas. Then the other common type would be epithelium. Epithelium, and this can be in the lining of the body. So this would be skin. So you can see that in the basal layer of the epidermis. That's where a lot of the skin cancers come from. Also the lining of the gastrointestinal tract, whether it's the cuboidal cells or the columnar cells in the intestinal stomach, or the pneumocytes in the alveoli or the bronchial epithelial cells. These are at risk for cancers in the lung. These are what we call a non-small cell carcinoma, or even in the breast. So this would be ductal cells or luminal cells that help with the passageway of milk. Again, this is a common location for breast cancer. Or you might also have it in epithelium that are secretory cells. So these are epithelium that produces substances, whether it's enzymes or hormones. They can also become cancerous. That could be seen in prostate, but can also be seen in breast and also see in lung. So this is just trying to illustrate to you that Labile cells are the most risky cells because they are the ones that are most mitotically active. Therefore, these are the ones that can generate mutations, which can then lead into an uncontrolled growth or tumour, which then can become malignant. Now, with that said, let's go through the most common cancers that we are likely to see. In the world generally, so this is from this data from the WHO, the top 5 most common cancers by incidence in the world is lung cancer, number 1, followed by breast cancer, bowel cancer, prostate cancer, and gastric or stomach cancer. Compare that to the developed world, it's breast cancer as number 1, followed by the prostate, lung cancer, bowel cancer, and melanoma. Now, so there is some similarity, but there is some difference. A lot of the difference comes down to genetics because genes can be inherited, so inherited risk of mutation can increase the likelihood. So there's certain things that we can't change. We can't change what we've inherited from our parents. Another big risk factor associated with cancer is age. As we get older, there's greater likelihood of mutations occurring, but also mutations being retained. And then the more mutations we get, the more likely cancer is to generate. And also our ethnicity is also a common risk factor. These are 3 that we can't change. They're non-modifiable. But there are a lot that we can. So some environmental risk factors. Number 1, well, at least for lung cancer, is smoking. Smoking is the greatest risk for lung cancer by far. Probably 90% of all lung cancers is due to smoking. That can be first-hand and second-hand smoking, as can air pollution. Air pollution is also a huge risk factor in particularly the developing world. So exposure to certain chemicals in the air increases the likelihood of lung cancer developing. Moving to breast cancer. Breast cancer, also smoking is quite a significant one, as is obesity. With obesity, it's important to note that adipose tissue isn't just a tissue that stores calories for another day. Adipose tissue is also a tissue that secretes hormones. Certain hormones can be cytokine-like, and cytokines are actually growth factors, and they can actually promote the growth of certain cells. So that's where cancers can come in, but obesity can also promote chronic inflammation, and chronic inflammation can also lead to more mutations and greater growth. So that's where obesity comes into play. Another one is alcohol. Alcohol is also a big risk factor for a number of cancers, breast being one, bowel being another. With breast cancer, another one is reproductive history. So it seems that unopposed estrogen, so this is where we have estrogen with less progesterone. So usually you get estrogen and progesterone in pregnancy. So mothers that have had more children are a little bit more protective, particularly if they're breastfed, because the levels of estrogen and progesterone have been remained higher for longer. Whereas females that haven't had children or they've had a longer reproductive history, so early puberty, late menopause with no children or only a couple of children, they've got a greater risk due to the effect of estrogen as being a growth promoter or a growth factor. Prostate and bowel diet is a huge one. So diet, low fiber, low fruits and vegetables, high fatty meat is associated with it, particularly certain processed meats has a high risk associated with bowel cancer. Also, they're showing more and more that the biota in the bowel, in the gut, that can become carcinogenic. So the bacteria in the bowel can produce certain factors that are carcinogenic, and that can be exposed to certain foods, particularly processed foods. So the diet is also quite profound in both prostate and bowel cancer. Interestingly, in gastric cancers, this comes down to a bacteria, Helicobacter pylori, which is a bacteria that causes stomach ulcers or gastritis, so that's that inflammation again. So that commonly causes inflammation to the stomach, and that chronic inflammation over many years, decades, can lead to cancer formation. So that can be rectified. And then finally, melanoma is exposure to UV. So you can see a lot of those risk factors. These are all technically modifiable. So when we look at the top 5 cancers in the world, 1 in 3 of them can be prevented by mitigating these risk factors. So the cessation of smoking, better quality air, losing weight, not drinking as much alcohol. In terms of diet, improving more whole food, more fiber, more fruits and vegetables, less processed diet. In the developed world, there's more exposure to the UV, usually a combination with lighter skin, such as myself, and stronger UV radiation, and in conjunction with poor sun practices. So this is where people deliberately are trying to get tanned, and that increases the likelihood. And then with bacteria, that's associated with gastric cancer, and that can be rectified. So a lot of these, if they're tackled, the risk drops significantly in conjunction with the certain screening programs, so mammograms for breasts, blood tests for prostate cancer, bowel screening, skin checks. All these things can mitigate the risk for cancer developing. Now, the last thing I wanted to tackle, and this is probably the most challenging, is what certain qualities does cancers possess that makes them similar? It's important to note here that cancer is a diverse disease, meaning we've got all these different types, but we have further subtypes. And in each subtype, they have further subtypes. And all these subtypes have different genetic changes, different genetic alterations. And the genetic alterations not only happen in the DNA, but can happen in the epigenetics and the way that DNA is transcribed and translated. So it's very complicated, and we can't really go through all the subtleties of the gene changes that you would expect to say between all the types of cancers and their subtypes and their subtypes. It becomes very, very complicated. But what scientists or academics have devised is that all cancers share these hallmarks. So there's certain qualities that cancers do show that they need to have or need to possess for cancer to manifest. And these are known as the 8 hallmarks. Now, what I've done here is I've put it into a mnemonic to make it a little bit easier for you to remember. So the mnemonic is miasma. Now, miasma is a term that means a noxious or a toxic environment. So this is probably fairly good for this to generate cancer, because cancer would have to occur in not a great environment. And so this is a good way to remember the hallmarks associated with cancer. So I'll go through these steps and then I'll explain them in a bit more detail. So the I stands for inhibition of growth suppression. I'll just write it down first and then we'll go through it. Next, S is self-sufficient. Next is metabolism. Next is immortalized. Next, A, angiogenesis. Next, S, suicide. Next, M, metastasis. And then lastly, A, avoids immune system. Okay, so let me go through them all now for you. So we've got the inhibition of the growth suppression. We've got the cells become self-sufficient. They change their metabolism. They become immortalized. They generate their own blood vessel, which is angiogenesis. Suicide or apoptosis, they lose that ability. They become metastatic, which means they move around and they avoid the immune system. So let's go through them one by one. The first thing they probably will do is become self-sufficient. And the term here is they develop a whole set of changes in genes, which we call oncogenes. Oncogenes are genes that are regulating the cell cycle to be turned on and permanently turned on. Normally our cells have control, so over here in the labile cells, they're controlled by proto-oncogenes, which are genes that regulate the cell cycle but know when to turn it on and turn it off. But these are being mutated, which changes the term from proto-oncogene to oncogene, which means now the cell becomes self-sufficient. They don't need to be instructed when to turn on, they just are constantly turned on. And this can be because they start to generate their own growth factors. So they actually make their own growth factors, which go back onto their own self and keep replicating themselves. They generate their own receptors, which means they become highly sensitive to growth factors. And ultimately, that just means you've got a whole host of mutations, which allows the cell just to be constantly replicating. So it's a bit like a car with its gas pedal permanently on. Now, opposing that, we normally have growth suppression. So there's certain genes, one you might have heard of is P53, which tells the cell when it needs to stop. So there are certain genes that are associated to be expressed when you want a cell cycle to be turned off. But in the context of cancer, we've inhibited those genes, which means now you've got a cell that's not only on, but it never turns off. So they're the first two really important mutations that need to take place. So a cell's on and the off signal is no longer able to be turned on. So now the brake is broken. So now the car is just pedal to the metal and nothing's going to stop it. The next thing we start to look at is the metabolic profile of the cells start changing. Normally, when we think of cells producing energy, like a muscle cell, we take in the glucose, we put glucose through glycolysis into the Krebs cycle, and then produce a lot of ATP. So we're getting 36, 34 ATP per glucose molecule with oxygen. But in terms of a cancer cell, it's not really concerned by that anymore. Instead of the cell being concerned about making lots of ATP from glucose, it's more concerned about making products to make more cells. So it shifts its metabolic profile to stay in glycolysis, so therefore make less ATP, but in doing so, it produces a lot more byproducts like more fats, more proteins, more nucleic acids. Why is that important? Well, it's all the building blocks to make more cells. So the metabolism changes of the cell, which means it's very glucose hungry, and it changes into being able to produce a lot more cells quickly. And so this is when we do a PET scan and we put a particular type of glucose with a marker, and we then look on a CT scan, we can see cells that are taking up lots of glucose. And this is why we can see cancer cells, is because the cell's metabolism is utilizing lots and lots of glucose. So it's glucose hungry, but it's making less ATP and making more building blocks to make more cells, which makes sense because its focus here is just to make more cells. So its metabolism changes. It then becomes immortalized. So usually what we see when we are a stem cell, like we see in bone marrow or epithelium, they've got a stem cell, which means they copy two daughter cells. One remains as a stem cell, the next one becomes a lineage that goes down a pathway. But eventually there's only so many passages these cells can go through before they become senescent, which is aged, and they become unstable in their mitosis, and then they will eventually die. What we see in cancer, they generate certain enzymes called telomerase, as an example, and it basically turns that the mortality off. And so it just continues to be able to copy and copy indefinitely forever, in conjunction with its ability to self-renew. So this is a bit like the properties we would see as an embryo. When we are in an embryonic stage, these cells have the capacity to kind of just keep replicating indefinitely. And this is the qualities that we start to see with cancer. They become immortalized, and that then means the cancer technically can continue to passage forever. And the next quality that cancer cells start to develop is a process called angiogenesis, which means the creation of new blood vessels. So once a tumor gets to a certain size, approximately 2 millimeters, it starts to run out of oxygen and nutrients through general diffusion, which means it needs new blood vessels. So the same thing takes place with cancer. As a tumor gets bigger and bigger, the inside of it will start to run out of nutrients and oxygen. Therefore, it needs new blood vessels. So what will happen is that certain cells will start to become hypoxic. They produce growth factors. Certain growth factors like VEGF, which is a vascular growth factor, which then encourages new blood vessels to grow into it. So new blood vessels will start to grow into it, supplying nutrients and oxygen and taking away toxic byproducts to supply the tumor cells with all the ingredients that it needs. Now, at the same side, this kind of feeds in with immortalization, is a cell normally has certain apoptotic pathways. These are cell suicide pathways to kill a cell when it feels like it's infected with a virus or it's incurred damage that can't be repaired. The cell will go through apoptosis to sacrifice itself for the greater good of the whole body. So what happens in cancer, and there's usually two pathways, there's an intrinsic and an extrinsic pathway. So with the cancer cells, they lose these pathways, so they almost shut them off, or they produce certain factors that make it anti-apoptotic, which means that the cell doesn't die. So all those genes that could be turned on when a cell is damaged or infected are no longer done in that particular way. So the cell does no longer partake in apoptosis and remains alive, and that helps with its immortalization. So no apoptosis. And it's usually through the intrinsic pathway. And that's the most common way that the mutations take place, is the intrinsic pathway is then switched off. Then we move to metastasis. Metastasis, which means the spreading of tumours throughout the body. The way it needs to do that, so when it's a cluster of cells in tumour, let's say an epithelial tumour, what it needs to do is it needs to break through the membrane below it, the basement membrane. Let's say if it's a melanoma, it's growing on the epithelium, but it needs to, if it wants to invade and go elsewhere, it needs to get through that basement membrane. So it needs to produce certain enzymes to eat through the collagen of that basement membrane. When it gets into that connective tissue layer with fat in the dermis, it can then migrate into the blood vessels. So it needs to invasation, which means intravasation, which means it grows into the blood vessel. So it grows through the endothelium into the blood vessel, which then incorporates itself with certain blood cells like white blood cells or platelets, and almost becomes like an emboli. And then it can travel through the blood, where it will then get stuck in another region and then leave the blood vessel, come out of the blood vessel. Then it can work back with angiogenesis. So as it's growing again in a new part of the body, it then tells new blood vessels to grow into it, so it can keep growing again. And that's where you see secondary tumours taking place elsewhere in the body. And that's ultimately through its metastasis. And then finally, we need to avoid the immune system, which basically means because these cells are becoming dysfunctional, the immune system is instructed to get rid of damaged cells or cells that possess certain qualities that might be neoplastic. But when it's a malignant form of tumour, it needs to avoid the immune system. So it needs to possess certain qualities that it can't be recognised. So the way it projects antigens is different. It's not self-recognised by certain immune cells. It can suppress T cells. It can suppress the overall immune system. So it can be very sneaky in the way that it invades the immune system. And this will be the future, or one of the futures of therapeutics, is kind of overcoming that issue and instructing the immune system to actually target immune cells, so your immune system can actually take out tumour cells in the body. Sometimes, so we're finished with the 8, sometimes they add an additional 2, which is 1, genomic instability, and 2, chronic inflammation. Basically what this means is malignant cells also possess a certain degree of genomic instability, which means it can change its genes very rapidly, which is important for its survival, because as it moves through the body, it needs to change its morphology. It needs to change its morphology to live in that new environment. So if it's a melanoma, which is suited for the skin, if it wants to go into the blood, it needs to change its morphology to live in the blood for that period of time. And then if it takes hold in the lung or in the bone or the brain, it needs to change its morphology to live there, because skin cells aren't meant to be in the brain or the liver or the bone or the lung. So having that genomic instability allows it to change its environment, to adapt easily. And that also speaks to when treatments are on board, like chemotherapy or radiation, that instability allows it to mutate, to overcome and become resistant to treatment. The last one is chronic inflammation. So what cancers will do is start to produce inflammation, certain cytokines, certain factors that are commonly associated with the immune system that causes certain systemic effects like anemias or cachexia, which is a weight loss. And that can also make the cancer worse. So by having chronic inflammation, it increases the likelihood for more mutations to happen, which exaggerates the whole problem. So that was a lot to take in, and it was a heavy video. It was a heavy topic to go through, but hopefully now you've understood your new terms. You can see the difference between a benign and malignant tumour. You can understand why certain cells are more prone to cancer than others in the body. We can see that there are common cancers both in the world and developed with a bit of overlap, but with the risk factors that are associated with it, as clinicians, how we can help our patients to kind of prevent cancers or mitigate cancers. And then finally, when we focus on the carcinogenesis, what leads to cancer, particularly malignant tumours occurring, they share these 8 hallmarks, which I've put here as the is-miasma, and hopefully that helps you to retain that. Thank you, and we'll see you next time.